Helene Steiner. I'm the co-founder and director of OpenCell, but I'll explain a little bit what it is. But I'm also the co-founder of Cell-Free Technology, what is a biotech company. And I have the pleasure to teach once a term the biomaterial platform in the fashion department of the Royal College of Art. And I thought I will actually start by sharing with you my first day at university. I was 19, extremely excited that I will study product design at the Bauhaus University in Germany. And I already pictured myself on the first day how I will sit in a studio, I will design chairs, tables, and shelves. And the first words of my professor changed everything on the first day, because he asked all of us if we think that we will be product designers after the course. And obviously everybody was raising their hands with full excitement. It was the reason why we were all in the room. And his response was, not even half of you will be. And it's not because you're not very skilled, it's because over these next years you will learn so many new topics, you will get introduced to so many new people, and you will get an entry to do so many new fields, and you should take the opportunity and make advantage of that. And it will change your pathways. And 65% of the jobs students actually will have, they don't even exist yet. And you have the chance actually to define those. And Sheila was actually pointing that already out. And I took the chance. So after studying product design, I actually moved to London and made my Master of Science in Imperial College, and also a Master of Arts in the Royal College of Art in Innovation Design Engineering. And the reason was because I wanted to understand how to integrate new technologies in my work, in particular electronics. And at the same time, I also received a very unique opportunity to collaborate with the MIT Media Lab and MIT Chemical Engineering, where I learned something I had absolutely no clue about beforehand. And it is working actually with living organisms, so integrating bacteria into the materials in the work that I'm doing. Which led then to a position at Microsoft Research, what was again completely out of my comfort zone, where I worked in the Human Experience and Design Group and Computational Biology Group, what allowed me to start thinking, how can I actually use computation to use into my work? So as a result, for me, design, engineering, and science really naturally merged in my way of thinking. And I think the world is really changing. If we look back in the last few years, disciplines were still fairly siloed, and collaborations were still not practiced, and interactions between the fields not really seamless. But today, the walls between the disciplines and the fields are blurring. New fields start really to emerge out of this, and this is, I think, where the truly impactful innovations start to emerge. And I think it's essential to have someone with collaboration and communication skills, someone who is able to use different vocabularies of the fields and has the ability to, on one side, constantly develop new skills and to evolve with industry, but as well with research. And I think that design can be here the unifying vehicle to drive the best experience. And I think out of this, kind of a new designer started to emerge out of this, the fusionists. So my colleague at Microsoft and me, we always called ourselves a fusionist, if I can say it. And the challenge and the reward of the fusionist is the ability to communicate and comprehend and connect all parties through design. And I think if you look at the global challenges that lie ahead, they can only be solved by a collaboration of minds and vocations and a diversity of views. And I think it's really important that we start building more sustainable systems that are in balance with nature. I particularly got interested in how we can bring our environment into our decision-making process. And I began investigating how living plants function. And they are incredible organisms. Plants are constantly communicating with the environment, with each other, and within themselves. And they cannot move, and therefore they have to constantly adapt to the environment and surrounding to be able to survive. And one way how they are doing this is through electrochemical signals, but are caused, for example, by ion fluxes in each single cell. And I thought, like, hey, electrical signals, that is actually something that I have learned at university and that I'm very familiar with. So we started to capture those tiny signals from the surface of the leaf of a plant with a relatively simple tool we designed, and it's not too far away from how we actually capture our own biosignals. And we started to look in the signals the plant generated and started to identify patterns. And the fascinating part was that the electrical patterns were actually distinct to different abiotic and biotic stress, meaning temperature, if it is infected, the humidity and light in the environment. Which led us then to the idea if we can actually map those patterns to the different environmental causes and find a way to be able to translate and understand them. And actually, while I was working at the project, I was sitting in my office, always alone with my plants, and from time to time, the colleagues always came by and asked me if I'm still trying to talk to plants, which I absolutely did, I think. So here you can really see how we capture from our current device. I tap twice the little hair inside the trap, what is the sensor of a plant, and then you see the mechanical closing of the trap. And every time the plant is doing that, it's exactly the same signal. So during my time at Microsoft Research, then I wanted to push this concept even one step further. And I wanted to explore if there is a way to create a two-way communication between plants and human. So I'll show you a little video. So in this project, what we did, we used language as a human input and light frequencies as plant input, and translated with the help of natural language programming, what is a machine learning process, human sentences into different light frequencies and durations. So what you can imagine is that we're almost like similar to a Morse code, but we sent through a living biological organisms. And then we translated the plant's reaction back into something human readable. You can actually visit that project in the V&A from the 18th of May onwards in an exhibition what is called Food Bigger Than the Plate, but it also talks about circular economy and everything. And I know that might sound abstract in first place, but when a plant gets infested, they produce similar electrical signals to turn on chemical reactions inside them to protect themselves. And Project Florence is a first step to actually explore if we can turn chemical reactions in plants digitally on, which would allow us to minimize pesticides on a field. At the moment, we just randomly apply them 6 times a year, but if we actually would know when they're infected, we could minimize it to just once a year. And you might ask yourself why I'm presenting this at an event where it's about fibers and wool and textiles. Because I think it's very crucial to look at every single part of the supply chain. Sheep need to eat, we have to have sustainable farming, and it also has a lot of aspects that we can learn from nature to actually apply biomimicry. But I also wanted to show you the project because it demonstrates the incredible capabilities and functions of cells. Some compare them to like small little microfactories, you can almost imagine. With the nucleus in the middle, what is the brain, which contains the majority of the cell genetic material, which is in form of DNA. And DNA is the code of life. And instead of zeros at once, like in programming, we have 4 letters as a programming language. It's T, A, G, and C. And this incredible creature, for example, contains a very unique genetic code, which makes it produce a protein which produces this mesmerizing green fluorescent glow. And in the 70s, scientists managed to isolate the DNA, what is the called gene, which is responsible for this, and to reproduce it in another organism, a bacteria cell. And that is only possible because of this tiny little manufacturing plant inside the cell itself, which reads the genetic code, the T, A, G, and Cs, which then get translated into a single-stranded code, which forms a blueprint of a protein production. And then there's a tiny part called the ribosome what actually produces the protein that you want, the DNA that you edited. And the process of creating this biological circuit is called bioengineering. And the process of introducing the circuits or non-native genes into a cell is called synthetic biology. And this particular protein I have shown you is called GFP and has become one of the most important tools used in contemporary bioscience, allowing us to understand how cells move proteins around organisms and let us to understand how cells work and also how we self work. And it ultimately allows us to make the invisible visible. We can use them as markers to watch the movements and positions and interactions of other proteins and use them as biosensors to detect, for example, pollutants in the air. And even so we know all of that, we have no idea why the jellyfish itself actually is producing it. But those proteins can be as well extracted and purified, and not only be used for science, but to explore how, for example, we can create more sustainable dyes. So at the moment, the dyeing industry is one of the most polluting ones, and Sheila was already talking about it a little bit, polluting the water and also the animals that actually live in the ecosystem itself. This is, for example, one of many dogs in India that turned blue because of the waste that goes from the dyeing industry into the rivers itself. But bacteria cells would actually allow us to produce natural dyes which are not harmful for the environment, and process would be a little bit similar to brewing beer. And the film that is just playing is from a company called Pili. They are based in Paris, and they are producing bacteria dyes. And nature is providing us here with a countless amount of beautiful colors that we can actually work with. But proteins do much more than glowing and producing pigments. Proteins are the key molecular building blocks for every organism on Earth. They define your eye color, for the structure of your hair, and also the structure of wool, and carry oxygen around your body. So during my time at MIT and MIT Chemical Engineering, we explored how we can use them as a growing living actuator to create a responsive bioskin. So the film that you just saw is a garment that reacts to the various body temperature and sweat and supports the body by through controlling these tiny little flaps that open and close regarding to the various need. Meaning all these tiny flaps are closed when you start running because your body temperature is relatively cool, but when your muscles start heating up and you start sweating, all these tiny little flaps actually open up. But as soon as you stop running, for example, or doing sport, your body relatively quickly cools down. So it wants to actually keep your body temperature, and in those moments, all the tiny little flaps close again. And the technology that enables this are actually bacteria cells. They have this incredible ability to expand and contract depending on the relative humidity in the air. And through applying those then on a substrate material, another material that doesn't have the materials, we were able to create these shape changes. And those cells, they're called Bacillus subtilis, they're actually edible, and they're extracted either from fermented soybeans, they're also called natto, it's a Japanese food, or you can also find them in soil. So they're not harmful at all. And there's another beauty of it, they can duplicate themselves billions of times just overnight. So you can also really use them on industrial scale and to reproduce them themselves. The only thing what they need is actually sugars. So for me, it was really there also to rethink the future of distribution and manufacturing systems. But for me, the most exciting part was the sensitivity to its environment. Opens up completely new biological interfaces without actually having the need anymore to embed any electronics. So in comparison to Florence, we still needed electronics to understand what is going on. So in our case, we used our own skin as the interface that creates a direct interactions between a human being and bacteria cells. And to make that happen, in Project Biologic, we needed computation as an enabling tool to build that biointerface. So we built a software ourselves which enabled us to engineer a fully functional garment by combining the data of the female and male heat and sweat map and the expansion rate of the bacteria cells in combination of the substrate we actually used. And the software creates as well a simulation and feedback loop of the designed material interactions to actually allow you to start designing it in the style you wanted. And connected with a hardware platform, we built a bioprinter. We were able to produce our ideas also in one-to-one and go away from just like a hypothetical idea and make it accessible for other designers and engineers and scientists. And enabled us to create many more applications because of that, from shape-changing lamps to animated origami to artificial plants. There were electronics together. And recently we published as well our further development in Science Advances, which demonstrates a garment which not only changes its shape regarding to the humidity, but also can start and stop glowing when needed. So very useful if you want to run at night to actually make yourself visible. And that is actually enabled by the protein that I just showed you beforehand. So what I have shown you so far is that we can take biotechnology further than for producing drugs or small proteins. And in the last years, we have started to understand how to make larger structures, like, for example, fibers. And I think most of you have worn at some point silk in your life, but I guess nobody really has worn spider silk. And Sheila already was talking about that it has these incredible properties. And the reason is why you haven't really worn any spider silk, because spider farms who exist, it's just not efficient enough to produce any garments out of it. But the protein itself you can extract and produce in bacteria, what has incredible properties, which can be produced either in yeast or bacteria with the help of bioengineering, which would allow you, for example, to create very strong materials that steel, they're biocompatible, so you could even integrate it into your body yourself, and they're biodegradable. So Jair Essaida, a designer from the Netherlands, for example, created a bulletproof skin made of spider silk and founded the company Inspidery, which is developing sustainable materials, but as well cruelty-free alternatives to leather. So you see how there's actually a bullet going into the material that she created. And actually, we have Nikki as well sitting here. I had the great pleasure that last year, Nikki James was in my biomaterial platform. And I really loved her project because she was looking into how hairs actually grow and how they get the shape and the properties from the structure itself. So if they grow straight out, they're actually straight, and if they are tilted, that's how they get the curls. And she was wondering if there's actually an opportunity to kind of like give fibers as well this function of curling up like hair when it's humid to create a sweater when it is cold and wet, who kind of puffs up to keep you warm, and when it is dry and warm, that it actually gets flat, that it's much more transparent. So Nikki hopefully can explain you a little bit more about her project, but I found it was a fantastic example to also see how can we now push those new fibers and apply new manufacturing processes to them to actually make them structurally much more relevant. And more companies and ideas like this actually start to appear, which engineer biology. But there's no too little space where you can go and build sweaters that are made of bioengineered organisms. So what I think is really missing as well in the ecosystem is a space where disciplines can meet and collaborate, and a space which gives you access to affordable lab workshops and studio space, and a place where critical thinkers and collaborative designers appear out of it. And because we couldn't find a place like this, we decided actually to build it ourselves. So Tom, who is sadly not here today, my co-founder, and I decided to move to London to found OpenCell. And I'll show you a little video how that actually looks there. So I'm Helene Steiner. I'm a design engineer, and I focus in a couple of years how we can combine science, engineering and design and look at how it might change our economy, our products, and how we actually might work interdisciplinary.
All the non-sustainable processes that we have today, we need to move away from because it's just not going to work. And so bringing together, young entrepreneurs with designers and the technologists and biotechnologists to think about how do we make things in a much more sustainable way? How do we create new materials? How do we create new products? How do we really innovate around this space? And this is what this space is all about, and it's bringing those people together. So OpenCell is in the end a space where we want to create an ecosystem and a critical mass to support biotechnology.
Instead of people talking about ideas or people who maybe have the capability of funding ideas wishing they would happen, I'd like people to actually just do stuff, kind of feel more able to take risks and push ideas out of research labs and into society. And lots of them will be losers, but every now and again there'll be something that's really useful and valuable.
What I'm interested in is the collaboration of the different ways that that thinking brings when you bring in a fashion designer earlier in the process. And then it's not that one leads, there's more of an interweaving, and that's when we're going to see that the form changes. Maybe bio will lead the aesthetic of fashion design, or maybe fashion might potentially insist upon what the science could be and where it should go.
Because for now I see this movement coming from like singular designer or singular scientist, but it's not there yet for big companies to really appreciate and to really merge this in something that it will be real.
I think the container labs could be a really good enabler. The cool thing is that you make something small, maybe also mobile, and that people who want to do science but not stay in academia or go to university can be creative and try to create something new.
We're living in this weird sort of time where we have tools that in principle would help people anywhere in the world to connect and communicate. And I think engaging the wider public in research is an exercise in constructing a world together, I would say. It sounds a bit hippie-esque, but that's how I see it.
So I guess I can hardly summarize that better than the people that have just spoken, but I would like to show you 2-3 tenets that we actually have in the work that is happening there. We have a lot of companies who are particularly looking into sustainable materials and in biomaterials to look at a circular economy. So one company is called Chipsport, and they are making a material that is 100% made out of potato waste. We're producing a massive amount of potato peels every day in London, or just in the whole world actually. So they're working together with McCain, and they get the whole waste material and figured out a particular fermentation process where they can turn it into an alternative to bioplastics, MDF and chipboard, but are all normally materials that you can hardly recycle and definitely not degrade. And that is, for example, the buttons that they make would look a little bit like horn. And another company that we have is Biome, who actually use mycelium as their material, and they use it as insulation material as an alternative. And what mycelium is, as Sheila was explaining that already a little bit, those are actually the roots of mushroom. And they are feeding from waste, so you can feed them the whole cellulose leftovers. So you could actually also really looking into recycling textiles there as well. So we have one open resident who is looking into the circle, how you can create it. But mycelium is really interesting because it has quite special properties. So they are particularly using it as an insulation material because buildings have to be very safe. So I guess we all know about what happened at Grenfell Tower. If you don't have the materials there that they are appropriate. And mycelium, what it does, it's self-extinguishing. So they gave one little part to BBC where they wanted to test if that is true. So they put a chocolate bar, a frozen chocolate bar on top of it, and then started burning it with a Bunsenbrenner over 1000 degrees Celsius to see if it's true. As soon as they turned off the Bunsenbrenner, the material was still as it was and the chocolate cream was frozen. What I think is not only interesting for buildings, but as well, I think, for clothing as well. Thanks a lot.